Namaste students, I am Jeevan Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Women's University, Jaipur. Today I am going to discuss about the topic ion exchange chromatography, which is related to the some separation techniques, which is chromatography. It is a type of chromatography separation. So in this video, I will discuss about the mm, basic introduction and principles and how ion exchange chromatography is conducted. So ion exchange chromatography in this technique it uses charge stationary waves to separate charge compounds. Means some charge periods are used in this regions that carry charged functional groups which interact with oppositely charged group of the compounds to be retained in ion exchange chromatography. What actually happen? We know that first we need to know about the basic thing. We know that if we have something which is negatively charged, if we have something which is have negatively charged, and that negatively charged particles will bind to a positively charged. It will not bind to any negatively charged particle. It will always bind to a positively charged particles. So what actually happens? We have added a stationary phase of negatively charged particles. Then it will be used for the separation of the positively charged product. Okay. If we have used a positively charged uh, stationary phase or beads, and so it will be used for the uh, separation of the negatively charged product. The product may be any kind of protein, any kind of enzymes. So we can separate these uh, products by ion exchange chromatography. So what is the stationary phase in the ion exchange chromatography? So the stationary phase is charged particle. It is a charged particle, and the charged particle is provided a charge, and the charge is conducted by a buffer molecule. Charge is retained by a buffer molecule, which provides support so that the charge may remain. So discuss about ion exchange chromatography in detail. The stationary phase. What is the stationary phase? Stationary phase is the phase from which sample is loaded. So the stationary phase in the ion exchange chromatography is a solid, and the solid in which we use in ion exchange chromatography is beads. These are the charged beads, and we can separate all these particles, all the particles by these charged beads. So the stationary phase is a charged particle which is solid, and the example of the stationary phase in the ion exchange chromatography may be beads, cellulose, and agarose. So these all can be used in the ion exchange chromatography as a stationary phase. And the next thing is mobile phase. What is the mobile phase? The mobile phase in ion exchange chromatography is a liquid. It is a liquid, and what kind of molecules are used in mobile phase? We use some buffers. We know what are the buffers. Buffers are the some liquid which have a particular kind of pH. They have particular pH, and whatever the pH may be, acidic, basic, but they have a pH, which provide support, to the electric field, so that the electricity can be flown by these uh, buffer molecules. So this is the liquid which provides support. They are considered as a mobile phase in the sense chromatography. What are the characteristics of the ion exchange chromatography? The separation of ion molecules and products are separated on the basis of their polarity and solubility and their charge. The main thing is charge, okay? And they are also separated on the basis of their polarity and solubility also. If the molecules have polarity to a particular kind of solvent, then uh, they can bind it very efficiently. If they do not have any kind of polarity according to the solvent, then their binding is not uh, good, and they will not bind properly. So separation will not be achieved in proper way. If solubility is also good, then it can be uh, important. The important thing is the charge. The charge, the molecules must be charged particles which we want to separate. So for example, if we want to separate proteins, so we know that proteins are both have both charge. They have positive charge and they have negative charge. They have both kind of charge. So if we want to separate the proteins of negatively charged, then we need a stationary phase. We need a weights of positively charged. For the separation of the negatively charged proteins, we need a weights of positively charged. So they will flow and they will bind to each other and can be easily separated. And similarly, if we have some positively charged, then we can use negatively charged beads. So they will be easily separated. What is the mechanism of the ion exchange chromatography? Charged molecules attached due to the positively charged stationary phase. Okay. The charged molecule will attract each other. Okay, and the attraction is between the oppositely charged stationary phase. Okay, and then as a result the molecules are separated and molecules of interest can be eluted by the adding another compounds which has more affinity for those charged particles. So this is the basic of the separation of the particles by ion exchange chromatography. What are the advantages of ion exchange chromatography? And the ion exchange chromatography have more matrix tolerance, sensitivity, and high quality product are obtained. Two advantages. First one is more matrix tolerance, sensitivity, and high quality product obtained. What is the disadvantage? It is expensive and has less reproduction, not a much big productivity is obtained. The iron. So some more things which we can briefly describe here. Iron is an atom or molecule which has lost or gained or more valence electrons. What is iron? Iron is an atom which has either lost or gained one or more valence electron. If it has lost, then it will be positively charged. If it has lost the electron, then it will be positively charged. If it has gained the electron, then it will be negatively charged. So on the basis of ion losing electron lose and electron gaining, they can be positively or negatively charged. So anions, what are the anions? Anions are the negatively charged particles. Cations are the negatively charged particles which are formed when an atom gain electron. Okay, when a atom gain an electron and anions are negatively charged because the there are more electrons. There are more electrons than the nuclei. Okay, there are more electrons in the protons in the nuclei. So they are more, they are negatively charged. Cations, cations are the positively charged ions, and they are formed when an atom loses electrons. Okay, when it will lose its electron, then the nuclei must have more, more protons than the nuclei must have more protons than the electrons. So it will be positively charged. So this is the basic of the ion exchange chromatography. I must also explain it with the diagram here. So this is a diagram of the ion exchange chromatography, and this is a example. The positively charged, here are some molecules which are positively charged, and some products are of negatively charged. So we have beads of both charges. Some positively charged beads are also there, and some negatively charged beads are also there. So both kinds of beads will be there for the separation of the charged particles. So the positively charged proteins will bind to negatively charged beads. Okay. If the proteins have a negative charge, and the buffer will flow here, and along with the buffer, the proteins proteins will also flow. So when the protein will come in contact with the beads, they will bind to the positively, some negatively charged proteins will come in contact with the positively charged beads, and they will bind to each other. Similarly, if there are some proteins which are positively charged, then they will come in contact with the negatively charged beads, and they will bind to each other. So two kinds of groups will be formed in this ion exchange chromatography. One is the one with the one of the group with which contains negatively charged proteins and positively charged beads. Okay, so they will be separated, and another group which contains positively charged proteins and negatively charged beads, so they will be separated in another portion. So by separation of these two portions, we can easily differentiate, uh, we can easily separate our desired proteins which have a characteristic uh, charge. So by using this method, we can separate our desired product, whatever the product is, a protein or enzyme. So this is all about the ion exchange chromatography, in which I discussed about the basic of the ion exchange chromatography, and the stationary phase of ion exchange chromatography, mobile phase of the ion exchange chromatography. What is the characteristic of um, separation basis of the ion exchange chromatography, and what is the mechanism by which molecules, proteins are separated, enzymes are separated by the ion exchange chromatography. Mostly it is used for the separation of the proteins and some enzymes also. I also explained the mechanism of separation, advantage of ion exchange chromatography, and disadvantage of the ion exchange chromatography. So I hope the students will better understood all these things. What kind of questions can be asked? According to this lecture, the question which can arise: explain chromatography, explain ion exchange chromatography, explain different stages of the ion exchange chromatography. What are the stationary phase and what are the mobile phase in the ion exchange chromatography. Explain the mechanism of separation by ion exchange chromatography and what is their applications. So these questions can be asked. So I hope students will better answer all these questions. So my lecture is over here. If you have any query, you can ask me.